Hello and welcome to the classes of Mastering Biology. Let's learn biology in an easy way. So today I will take up the topic nephron, which I have already told you that it is the structural and functional unit of the kidneys. Why is it the structural and the functional unit of the kidneys? Because it helps in filtration of the blood. All right. So first we will talk about the structure of nephron. Now when I say the structure of the nephron mainly comprises of two parts. The Malpighian corpuscles and the renal tubules. Now if you remember, I have already told you why are we using the word renal. The word renal will always be used when we talk about the kidneys. Malpighian corpuscles further has two parts, the glomerulus and the Bowman's cup. These are the two parts. What is glomerulus? Glomerulus is the tuft of capillaries where filtration of blood takes place. After the filtration, the filtrate needs to be collected. For that purpose is the Bowman's cup, which collects the filtrate. All right. So this is the tuft of capillaries and this is the cup and it is present like this. So filtration takes place here and the filtrate gets collected in this cup. Next, the renal tubules have three parts. The PCT, known as proximal convoluted tubule. The Henle's loop or the loop of Henle, which is a U-shaped tubular part, and the third one is the DCT. Full form is the distal convoluted tubule. Now, why the term convoluted is used? The word convoluted means it is coiled structure. Coiling is to increase the surface area for the respective functioning. All right. So this is the outline of the structure of nephron. Now let us quickly draw the nephron. Yes, we will draw the nephron and it is very, very easy. Nephron is closed at one end. It is a tubular structure which forms a cup-shaped structure at one end. That means this is the closed part. This is the Bowman's capsule. This is the glomerulus, which is the tuft of capillary, collectively forming the Malpighian corpuscle. This collectively forms the Malpighian corpuscle. Now, this structure leads into a coiled part which is known as the PCT. I'll draw it a little up here. This is the PCT. and then forms a U-shaped structure, which is the loop of Henle. And then we have the DCT, the distal convoluted tubule. And it ultimately leads into this narrow structure, which is called as the collecting tube. The collecting tube opens into the main collecting duct. All right. So this part is the PCT, the proximal convoluted tubule. This U-shaped part is the Henle's loop. This one is the DCT, the distal convoluted tubule, and this leads into the collecting tubule, and the collecting tubule from various nephrons open into a common collecting duct, which leads to the outside of the kidneys. All right, so I hope I'm clear with this part. I'll just quickly repeat the structure again. This is the glomerulus, the tuft of capillaries, which is held inside the cup-like structure, which is called as the Bowman's capsule or the Bowman's cup. All right. The Malpighian corpuscle is the collective term we use for glomerulus or the Bowman's capsule. Then we have the PCT, which is a highly coiled tubule. Then we have a U-shaped structure, which is called as the Henle's loop. Then again a coiled structure, which is the distal convoluted tubule, which opens up into collecting tubule and finally opening into the collecting duct. And from this collecting duct, that means after all the waste has been eliminated from the blood and finally when the collecting duct leaves the kidneys, now this filtrate is called as urine. And this urine is then passed into the ureters and then into the urinary bladder following the excretory pathway. All right. So this is the basic structure of the nephron. And here we will also draw the blood capillaries that are entering. So this one comes from the branch of renal artery, and this is the afferent arteriole. Why afferent arteriole? Because this blood vessel is bringing the blood towards the glomerulus. So this term afferent is used. After the filtration has taken place, all right, we'll discuss the formation of urine separately. But here you need to remember, after the filtration has taken place, most of the content of the blood is filtered out. But some of the parts, some of the proteins which are large enough, they cannot be filtered out and they remain in the blood itself. So they are carried to the outside of the Bowman's capsule by the efferent arteriole. And this efferent arteriole term is used because the blood is being carried away from the glomerulus, from the glomerulus. All right. This efferent arteriole then branches to form capillaries around the DCT, and it further branches here to form a network and this network of capillaries around the nephron is known as the vasa recti. This is the vasa recti, the branch of capillaries which is present around the complete nephron is called as vasa recti, and this opens into the branch of renal vein. Now, if you remember, when we had understood the human excretory system, I told you that the renal artery enters the kidney, and then we also drawn the renal vein leaving the kidneys. All right. So renal artery, when it enters the kidney, it branches into number of arterioles. Each arteriole enters one glomerulus, and from that arises an efferent arteriole, and then ultimately ultimately the blood gets deoxygenated and it's filtered at the same time, and then it is collected here by the renal vein and sent to heart for oxygenation again. All right. So this is network of capillaries which is present around the nephron is called as vasa recti. So to tag the figure, you will write structure of nephron. This is the structure of the nephron. Now let's talk about the functioning part, how the functioning takes place, which is the physiology of urine formation. The physiology of urine formation. Urine formation takes place in three main steps. The first one is the ultrafiltration. The second one is selective reabsorption. And the third one is tubular secretion. Now let's quickly get back to the diagram first, where we will understand the concept of these three steps along with some more structural details. All right. And then we will talk about them. We'll write about them here. So when I'm talking about the structure of the nephron here, the glomerulus, the Bowman's capsule, the blood is filtered here. The blood which is filtered here is under high pressure. So that was the first step, ultrafiltration. Filtration is normal sieving, but when it takes place under high osmotic pressure, then it is called as ultrafiltration. So because of that pressure, the first step happens here in the glomerulus, which is the ultrafiltration. I told you almost all the components of the blood are filtered out, even the useful material, except for some large proteins which are then passed into the efferent arteriole. But now this filter, the filtrate gets collected here in the Bowman's capsule. All right, the filtrate gets collected here in the Bowman's capsule. This filtrate now moves down into the PCT, where the second step of urine formation takes place. That is, all the useful materials are reabsorbed. Absorption of glucose, amino acids, certain salts, these takes place here in the PCT. As the filtrate moves further in the loop of Henle, here reabsorption of water takes place. Maximum amount of water is reabsorbed here in the loop of Henle. And as we proceed in the DCT, what is going to happen here? Now there are certain waste which are still present in the surrounding cells and the surrounding tissues, and they have not entered the bloodstream yet, but they need to be removed. So to get rid of this additional waste, the third step takes place here in the DCT, which is the tubular secretion. So these are the three steps of urine formation. Ultrafiltration, which takes place in the glomerulus. The filtrate gets collected in the Bowman's capsule. The filtrate has both the useful and the non-useful substances. The useful substances get reabsorbed here in the PCT. Absorption of water, the reabsorption of water takes place in the loop of Henle. Then as we proceed further in the DCT, additional waste are transferred in the filtrate. All right. This is the third step, which is the tubular secretion, and ultimately the waste reaches here in the collecting tubule and then the collecting duct. And when this collecting duct opens outside the kidneys and the filtrate now enters the ureters, this will be called as urine. So unless and until it is inside the kidneys, the filtration is taking place. There the term we used is the nephric filtrate. Why nephric filtrate? Because the filtration is taking place in the nephron. All right. So let us quickly write a few points about these parts, and then I will make you write the functioning, the physiology part also. So here we've already written that glomerulus is the tuft of capillaries. Bowman's cup or Bowman's capsule collects the filtrate. Then the PCT, the proximal convoluted tubule. Let us note a few points here only. The PCT is highly coiled tubule. It has microvilli on the free surface. Why it has microvilli on the free surface? To increase the surface area for absorption of the useful substances. All right. Then Henle's loop is a U-shaped tube. Here, reabsorption of water takes place. All right. And the third is the DCT, which is again a coiled structure, a coiled tube. Which opens into the collecting tubule. All right. So you can write this as a structure part for your exam in the form of the flowchart only if you want, or you can write it point wise. I find easy to understand and write in a flowchart manner.